panel. So I hope we have lots of good questions. I see dozens of people lining up with mics. Again, people online, the Q&A pod that's in the lower right-hand corner, go ahead and put your questions in there. But we'll start with the question here in the room. Nice and close to the microphone, please.
Thank you very much, and thank you to the panel for speaking. This question is directed towards Dr. Gaglani, and the question has to do with nasal solution applications products. And there was a question and answer on stability requirements put out a little while ago. One of the footnotes said that for nasal products, they wanted to have 3 lots of API. And I was just wondering, how would that be applicable to nasal solution stability? And is it a requirement for nasal solutions?
Nasal solutions, and again, and I understand there's a low risk as compared to the suspension products. But if you could use 3 lots, we recommend that. But we also rely on other data as well. I mean, if you have some other data, in vitro data to demonstrate that, then that should be suffice. But we do recommend, I mean, typically we do recommend that 3 lots be used for the nasal products as well, especially for suspension product.
All right, microphone number 2, nice and close for me, please. Oh, let me turn that on for you. Sorry. We're conserving batteries. There we go.
Hi, this question is for Dr. Walenga. So first of all, thanks for a very interesting talk. The question is, and so I assume that with diseased versus healthy lungs, you have very different phenotypes in terms of resistance and flow rates and eddy currents and whether there's turbulent flow in the lungs. And so were these models developed with healthy lungs, I assume, and do they extend to these different disease states or COPD, fibrosis, and can they be applied in these different disease states?
Yeah, thank you for your question. So the models that I was showing there were healthy models, but we have done some work, and not just us, but other groups have done work on developing disease models. So the figure that I showed for small airway deposition from the University of Iowa, so they've been developing several different asthmatic models, which are based using a cluster-based analysis. They collected data from 250 subjects, and they're grouping them into functional clusters. So they're developing models for asthma, and then they're going to be beginning work on COPD models as well. So specifically with narrowing the airway diameters.
All right. Thank you much. Can we have our first online question, please?
Yes, this question's for Dr. Conti. In some product-specific guidances for OINDPs, the FDA recommends performing in vitro studies at BME life stages. However, for capsule-based DPI products such as Spiriva HandiHaler, how do you differentiate the life stages?
So the question is regarding life stages for capsule-based dry powder inhalers. So in this case, the way we understand is that the device comes with a certain number of capsules. So depending on the number of capsules, we define beginning, middle, and end life stages. So suppose the package the patient gets contains one device and 50 capsules. So the first capsule we identify as the beginning of the life stage of the product, capsule number 25 middle, and at the end capsule number 50 the end of the life stage. So we basically consider the device functioning throughout all the capsules that are supplied to the patient.
Okay, microphone number 2, nice close for me, please.
So this is a question for Dr. Conti. Before I ask the question, I'm not seeking comment from FDA regarding what other global regulatory authorities are doing. But to give a perspective, EMEA for inhaled drugs require in vitro PK studies. If not favorable, then go to PK. And then, if not favorable, go to PD. Health Canada, same thing, in vitro PK or in vitro PD. FDA requires all the three studies. So the question is, in the light of real-world evidence and real-world data, will FDA be revisiting the three-study requirements in order to determine whether these requirements are overbearing, or could a regulatory reprieve be offered?
So this is basically a question regarding differences in terms of regulations across the countries.
Yeah.
We understand that European countries and other countries, they have different regulations compared to US. Currently, for these complex products, as we discussed here, we believe that the current thinking is relying on the weight of the evidence approach in terms of demonstrating bioequivalence from all components, in vitro, PK, and clinical endpoint study, in addition to formulation device. We understand there are difficulties, especially to the clinical studies. That's why we basically welcome alternate bioequivalence approaches. I think in general, our guidances are not binding, and we are welcome to discuss alternate approaches for a specific product. As I mentioned in my presentation, those products, they can be very different. So it's difficult to us to establish one pathway for that would cover all products at the same time. So that's why we rely on the weight of the evidence approach, and we basically look into case by case. And then I think right now, the GADUFA 2 program, we have a very solid communication pathway, so then industry is welcome to come to us and then discuss alternate approaches.
Thank you.
And I know we've got something in the back of the room. Please add it. Since this one, again, we're trying to keep the questions less about policy and future decisions of the agency, more about the science that the folks are presenting on. So we'd appreciate you reining those questions into what's being discussed today, not trying to bring the agency to make some policy decisions in a forum where it's not going to happen. But I know our SMEs had something they wanted to add in the back of the room.
Hi, this is Kim Witzman again. So I did want to say I agree with everything that Dr. Conti says. I think the point is that our guidances are not binding. If you have an alternate approach, then we do welcome pre-ANDA meetings to discuss those approaches. And I think ultimately the question really turns on the first sentence, which was that EMEA has a different regulatory paradigm. So we have a regulatory paradigm that tells us that we need to define sameness of the product to its reference listed drug. EMEA does not have the same approach, and there are ways that they can demonstrate their equivalence or hybrid models which are not the same as the US regulatory standards. So therefore we have to work what we have, and EMA can do what they have.
Thank you. And if there's other questions in the room, please come up with the microphones, but let's take our next online question, please.
This is for Dr. Ma. How does the agency recommend applicants present or justify sameness for a combination product when they see the RLD has more variability than that seen for the proposed test product, such as during a bioequivalence study? For example, nasal spray in which the RLD spray pattern has a wider range compared to the test product.
The in vitro study that this questioner asked about, I think because The bioequivalence was for spray pattern study was based on the population B test, and having wider RLD variability actually make the PB analysis easier to pass. In addition, so this is for spray pattern study in general, and if I think as long as the test product meets all the B requirements, as compared to RD product in terms of in vitro test or PK test or PD test or clinical endpoint study test in general, then it will be recommended for approval.
All right, thank you. Microphone 2, please, go ahead.
Hi, this question is for Dr. Ma and maybe also for Dr. Lee. We are talking about the dry powder DPI product, and we have observed both in the reference and in some degrees also in test product. The first blister of the product and normally have the wider variability on the for example, SAC or APSD. So the question is, if we, normally if we do the in vitro BE or the in vivo PKBE, we use the first blister. So in this case, because we have evidence of that, the variability, larger variability of the first blister, can we use the second blister for study?
I guess you mentioned the first blister is that pertaining to the in vitro study or?
From the in vitro study, we see the higher variability of the SAC and also APSD. So, and also the amount of the drug is also lower than the rest of the blister. So, the SAC is the amount, and APSD and the total amount is also lower always for the first blister.
So because the in vitro test for SAC and APS, so for APSD it was recommended for the beginning, middle, and end, and APSD is recommended for the beginning and end. So as long as the blister that you use meet the beginning life stage definition, I think in general we will be okay with that.
So yeah, if we choose in this case, if we have the data showing that always demonstrating that first blister, for some reason we don't know yet, but for both test and the reference, we found that trend. So we can use the second one, but no, second one is always okay.
Yeah. Sorry, what's your question again?
No, my comment is that we don't find a problem with second blister. So it's always the first blister. As you just responded that the beginning stage, we could consider second still at the beginning stage.
Yeah, as long as meet the definition of beginning life stage.
Okay, thank you.
Okay, thank you.
All right, let's take another online question, please.
This is for Dr. Spagnola. Do you think it would make sense to put your specific needs with regards to rescue med tables and how this is presented in the PSG?
Thank you for that question. I think if I'm understanding it correctly, they're asking about, why doesn't the product-specific guidances include information about rescue medication? And I think it's important to just emphasize and reinforce that the product-specific guidance are just our recommendations. They're non-binding. And so, it's important that there are important things to define in the study design and in the study protocol that are not included in the product-specific guidance. These are not supposed to include everything in the study design that you should take into consideration. And so that's why we have forums like these to help educate and recommend things. And so I think hopefully understanding that it's up to the applicants and sponsors to predefine and mention those things in the study protocol, even though they're not defined or outlined in the product-specific guidances.
Okay. Microphone number 1, nice and close for me, please.
This is a follow-up question, actually. This has been brought up a couple of times about lot-to-lot variability of the batch, stability batch. So the question is most for Deval of the panel as well. So earlier there was a question about for the stability batch, we expect 3 lots of APIs. What's the agency's expectation on the device and acceptance, do we require additional lots from these components? And additionally, for stability batch, for long-term stability, what the worst case scenario will be sufficient? We just put that on the horizontal position. If we define this as the worst case, would that be sufficient to meet expectations?
Thank you for the question. I think that was one of the things in my presentation. As far as the lots are concerned, we do recommend that at least for the device, since it's a critical constituent part for the combination product, so and it can results arise in a lot of variability because of the device. So we recommend that the registration batches not only be manufactured using 3 API lots, but also device, including if you can, especially for an excipient which plays a critical role for dry powder inhalers, for example, then you want to ensure that also you include 3 lots of lactose as well for your product for registration batch. The second question which you had was about
the orientation of stability
right.
batch.
So as far as reorientation is concerned, we do recommend that to understand the variability of the drug product on stability, that at least minimum 2 different orientation be studied. So yes, I mean, unless you can provide that information that going forward, I mean, during the registration stability that that the given orientation is the worst scenario, then going forward you can do one orientation itself. However, for registration stability, we require at least 2 different orientations.
Thank you.
Right, microphone number 2.
A science-based question for Dr. Vaninga. Typically, the airflow rates in the trachea and bronchi are very turbulent, but computer models base it on a laminar flow. What was the type of flow that you had used for CFD? Approximately the Reynolds number that you observed.
So I think there are 2 parts to your question. So the first part seems to be, are these models laminar based? So they're only laminar based when you, as you mentioned, the Reynolds number is below a certain value. So for, I think all of the models that I showed, they're all using either a 2-equation turbulence model or a large eddy simulation model. So that they do mostly consider turbulence. The Reynolds number could be, say, 5 to 10,000 at the entry. So it can be quite high.
Yes. Thank you.
This is for Dr. Lee. Based on your presentation, is the FDA considering widening BE acceptance limits for PK metrics?
Thank you for the question. This is something kind of controversial in the literature, and also for you may aware of some of the citizen petitions submitted to FDA regarding the similar questions. In the response to those citizen petitions, the agency did not comment on the acceptability of the widening beam limits or alternative approaches such as multiple batch PKB studies. And the approval of the ANDA with such an approach will depend on the justification, also depend on the information available to FDA, so then FDA can assess the data and then can make a revision of the PSC if properly implemented or scientifically sounding. Thanks.
All right, one more online question, please.
Okay, this is for Dr. Wilenga. Is there a set of criteria that the FDA will use to evaluate CF models used to support novel bioequivalence approaches?
Thank you for your question. So I had mentioned ASME VMV40. That's just one way that you can try to either verify or validate your model. So we don't have a set criteria at this time. But that method is quite robust, so I would suggest looking at that one.
Okay, and that one was so quick. Let's online question.
Yep, this is for Dr. Conti. If a proposed generic DPI device has some differences from its RLD, as identified by comparative analyses, is the pre-ANDA product development meeting request the most appropriate way to communicate with the FDA?
So if I understood the question correctly, there are device differences, and the question is if the pre-ANDA meeting is the most appropriate way to communicate. I think first, after conducting comparative analysis as per the comparative analysis guidances and identifying the differences, if the questions are specifically on the device part, perhaps a control correspondence would be the first way to communicate. Control correspondences they have 60-day clock. So in 60 days, we can address the specific questions about the device. And if we identify additional issues, then further during the development program, we can discuss more in detail through pre-ANDA meeting packages.
Before we wrap it up, based on any of the questions you heard, any of the discussions we're going on, anybody on the panel have any closing thoughts they'd like to share before we go to break? Yeah, Ross, please. Push it one more time.
On the last question I got about verification and validation, just want to reiterate that if you have a method for validation and verification and you have a package you want to submit via pre-ANDA meeting, we would certainly encourage discussion.